If you're just starting this video, please make sure that you view the end of the previous video on genetic engineering with restriction enzymes, cloning a gene, and gel electrophoresis. I kind of ran out of time with the allotted minutes that I have per video. So this is where we left off, the idea that the DNA can travel through a gel, but it is somewhat invisible to the naked eye, so therefore we need a stain, and it's a fluorescent stain. You wouldn't do this in a typical class. In abbreviated versions of it, you might use just food dye or molecules that are visible, and you'd be able to see the separation. But this is the picture where they expose the fluorescence so we can see where the DNA traveled. And we'll get away from that, and then here is more of a labeled picture. So recall that the first lane, this is where the DNA traveled. So this first lane had with only one enzyme cut, so therefore there were two fragments. Second lane had one cut, so two fragments. And in the third, there was both enzymes added, so therefore each enzyme cut, which meant two cuts of the... Sorry for that. Sorry, there was an announcement in the background. I want to stop. The last lane here has three segments due to the fact that the DNA was cut twice, once by each of the enzymes. I'll explain what this lane is in a few seconds. But the premise of gel electrophoresis is that if they all start at the same point and they run for the same amount of time, the shorter pieces of DNA are going to travel further because they're smaller. They can get in between the areas much quicker. So fragment D would be the same size fragment. It's just been run in both gels, or both lanes, sorry. So D would be the smallest. It went the furthest. A would be the next smallest, or kind of the third largest. Here is, or sorry, the fourth largest. This is the middle, where it's in between in terms of size. I hope you get the idea that C would then be the largest. So that's just very qualitative. But if you want to know more specifics about a quantitative value, they can put a control group over here. So what they do is they add specific fragments of DNA that have known lengths. And here they have a variation from 560, the BP stands for base pairs, and that's just nucleotide sequences, so that'd be 560 nucleotides strung together. Then they have all these other variations. So what I can say is that if we look at letter E, what we have is letter E would be between 4,400 base pairs and 2,300 base pairs. More likely closer to 4,400 because it's closer, but we couldn't give it an exact. It just gives us more of a approximation of numbers as opposed to just E is smaller than B but is larger than A. So that's how a gel works. The application to forensics is actually used with things like paternity tests and identifying criminals that may have left things at the crime scene. So if we envision this process where they're trying to convict someone of a crime, what they would do is they would take a sample of evidence and maybe put that in the first lane. They expose it to a restriction enzyme, and then any suspect that they think might have left that there, they would take a sample of their DNA and cut it with the same exact enzyme. So if the DNA left at the crime scene was cut and it was made by the same person that's a suspect, if you use the same restriction enzymes, then what will happen is the restriction enzymes will cut the DNA at the same location. So therefore you will have the same number of fragments, and those fragments will be the same size, and they'll travel the same length. So we'd be able to tell if someone is the person that left the evidence at the crime scene if their DNA fingerprint is the same. But if someone was not the person that left the evidence, then the restriction enzyme would not cut in the same location, the same number of times, so you would see a different fragment picture over in their lane, and then you would kind of say, well, that person did not leave the evidence at the scene. Another application is paternity tests. So what I'll do is I'll put a picture of a paternity test gel electrophoresis, and we'll kind of talk about that. Here we have a sample gel that would have been run for paternity test. The people involved that you would need to know their DNA sequence would be the mother, the infant in question, and any suspected father. So here in this case we have two suspected fathers trying to find out which one was the father of the infant. Now based on knowledge of DNA, what you have to realize is that an infant is 50% mother, 50% father. So when you run a gel, the fragments that the infant possesses should also be a representation of 50% mother, 50% father. So if we look at this first fragment, we can see that this fragment is found in both the female and male number 2. Since we can account for this being part of the female, we can't really assume that this is the male yet. For the second one, we have the female could contribute, but also male number 1. So still, these two don't really help us. If you look down here at fragment number 3 that's numbered, we notice that the female does not have this fragment. So that means that the infant did not get the DNA sequence from mom. So therefore, the father would have been the contributing parent to this fragment. So notice that male number 1 does not have that fragment. So he can be ruled out as the possible father because there would be no way that this infant would get that sequence with neither parent having it. But it is male number 2 that has it. So therefore, we look at all of them. Every fragment that is on this gel for the infant has either one from the mother and one from male number 2. All right, there are some cases where it's both scenarios, and that could happen, but we're really looking for 3 is kind of the ideal situation where the mother could not have contributed, so the father had to. So that goes through the process of gel electrophoresis and 2 very common applications of it. So hopefully that helps you as you are studying this process.